All right, welcome back. We left off from our last lecture talking about osmosis. This is what happens when there is a concentration gradient that is caused by a solute that can't cross the cell membrane. So if you remember, we came back here. Let's look back here, where we put a cell in an environment. I'll just outline this cell one more time in a different color. There we go. Where there's a higher sodium concentration outside the cell. All these sodium ions. We talked about how if you ate a whole bunch of salty popcorn at the movies, you are bathing your cells in a very sodium-rich environment. So you have a high concentration of sodium outside the cell and a low concentration of sodium inside the cell. And that's a concentration gradient, and it's not stable. So instead of having, a concentration of 6 sodium ions outside the cell, these sodium ions a few of them want to move in, so that we could have 3 and 3, but that's not going to happen because sodium is not allowed to diffuse across the cell membrane. The cell membrane is a semi-permeable membrane, and it says, sorry, sodium, you are not on my list. Now, water, on the other hand, is allowed to cross the membrane. So, as you know, as if you recall from last time, there's a high concentration of, not a concentration of water, there's cytoplasm in the cell. This is water-based, so there's a lot of water inside the cell, and the water will actually exit the cell in an attempt to dilute out this sodium ion concentration outside the cell. Can you see that as I'm adding a whole bunch of water out here, that the sodium concentration will at least go down a little bit? Yeah. That's osmosis. Osmosis is the diffusion of water across a semi-permeable membrane in an attempt to equalize a gradient caused by a solute. So let's look at that definition that we got here. Osmosis, right here. Again, these are just all notes from last time. We're just rebooting a little bit. Osmosis, the diffusion of water, which is the solvent, across a semi-permeable membrane in order to equalize a concentration gradient caused by a solute. And we talked back here about how when you have a hypertonic solution is when the sodium concentration outside is greater than the sodium concentration inside the cell. And we talked about how water exits the cell to try and dilute out that sodium ion concentration. That's what we have here. Here we have a hypertonic situation here. There we go. And we also talked about how it looks, we talked about how, water's going to exit the cell dehydrates, and it could actually shrivel up. So cell shrivels and dehydrates. Okay, and we talked about how a plant cell in a hypertonic solution is a little bit different. We talked about how if you put salt on a plant, and we're like, well, why would anyone want to salt a plant? Well, we pour salt on the roads, and this is what happens to the plant cells that are near the road or who get an increase of sodium ions in the soil, in the nearby soil. Let me just erase this a little bit right here. There we go. Let's get back to light green. All right, we talked about how you have a rigid cell wall that has this very rectangular brick-like structure, and inside of it is the flexible cell membrane in the plant cell, and it's a phospholipid membrane. So it's very flexible. And what happens is that, okay, you've got a bunch of water inside the plant cell. Plant cells have water. And yeah, there's probably some water out here too in the environment. But if you place a plant cell in a hypertonic solution where there's more sodium ions outside the cell than inside, these sodium ions want to move in, and the cell membrane says, nope. Do not pass go, do not collect $200. So water's like, I can fix this. Water exits the plant cell, and that essentially dilutes out this sodium concentration that's outside the plant cell. Now, as that happens, though, let me erase the water here. There we go. As that happens, the contents of the cell membrane will shrink and shrivel. But the cell wall that's rigid will stay in place. So again, I can't remember if you had that lab yet, your diffusion and osmosis lab, where you basically put plant cells, that Elodia, remember Elodia? That's that aquarium plant where you rip the leaf off and you took a look at it underneath the microscope in a hypertonic solution, and you probably saw something that looked kind of like this, because it's the cellular contents that are inside the cell membrane that shrink up. But this rigid cell wall out here stays intact. So the cell wall stays intact. The cell membrane, that's this out here, the cell membrane and its contents shrivel up. So, and it looks really obvious under a microscope there. That is a plant cell in a hypertonic solution, right there. So a hypertonic solution is where the sodium content outside is greater than the sodium concentration inside the cell. Let's look at the opposite situation though, because it could happen. Let's see, let's pick a different color here. Go with this one. Let's go with red. All right. Actually, human red blood cells, human red blood cell, RBC stands for red blood cell. Human red blood cells, let me give it a little nucleus in there, whatever, actually have a bit more sodium in the cytoplasm than a lot of other cell types. So it was a little misleading of me way back here, hope I didn't give anybody a headache there, to show that there was no sodium inside the cell. There's definitely some sodium that is in the cytoplasm, but I wanted to demonstrate having a higher concentration outside than inside. But it's not always the case, and red blood cells actually do have a slightly higher sodium concentration than a lot of other cell types. So, let me put some sodium inside the cell there. Now let's say we put this red blood cell into an environment where it's just pure water. No sodium at all. Just water outside the cell. Oh, and remember, there is water still inside the cell, in the cytoplasm, but like no sodium out here. Yep, I gotta do that in the sodium color that I've been using here. Okay, so no sodium outside. So here we've got a high concentration of sodium inside the cell, but low or no sodium outside the cell. This isn't hypertonic. This is what we call a hypotonic solution. And remember, I'm abbreviating solution with SOL''N. I think I need a slightly pointier, there we go, finer point there. Okay, a hypotonic solution is when the sodium concentration outside the cell is less than, lower than the sodium concentration inside the cell. All right, so if we've got a concentration of 1, 2, 3, 4 sodium the concentration of 4 inside the cell and let's say 0 outside the cell, that is a concentration gradient. Is a concentration gradient stable? Nope. It is not stable. Nature doesn't like a concentration gradient. Nature does not favor a concentration gradient. It is not stable. Nature does not want 4 inside the cell and 0 outside the cell, so 2 of these are going to want to move out, right? But the cell membrane says, nope. You're not allowed to cross the membrane. I'm a semi-permeable membrane. You're not on my list. Whatever. It is just an observation that sodium cannot simply diffuse across the cell membrane. So it can't fix its problem. It can't equalize its own gradient. So water's like, I got to fix this again, don't I? But this time, water's not going to exit the cell. Think about what would happen if water exited the cell. It would make the concentration inside the cell even more concentrated. It would make the sodium ion concentration in the cell even higher. So that is not going to happen. Water's not going to exit the cell. This time, water is going to move from the cellular environment inside the cell. And can you see that as enough water moves inside the cell, that sodium concentration inside the cell will lower? Oops. But think about what would happen if water keeps entering the cell. What's going to happen to that cell? It's going to pop like a balloon, or it could. I mean, first of all, the cell's going to swell and it's going to look all bloated with water. And if enough water has to move in, like, let's say this is a very high sodium concentration inside and a very low concentration outside, the cell could actually burst and pop like a balloon. That's actually part of what happens with something called hyponatremia. If you want to look that up, let me see, how do I spell that? So this is an aside. As an aside, if you're curious, look up hyponatremia. I think that's how you spell it, hyponatremia. It can occur in extreme temperatures when a person is exercising very hard and really pushing a lot of water. It can happen. It's a little more common along with altitude sickness too. But basically, what's happening is that there becomes an imbalance in the body, and too much water can enter red blood cells and they can burst. Not a safe thing. And anyhow, that is very, very, very extreme. But it is common that plant cells and that some human cells can be in a hypotonic solution and just swell a little bit. So let's look at this hypotonic solution when the sodium concentration outside the cell is less than the sodium concentration inside the cell. Water enters the cell. The cell swells, or I don't know, you can call it bloats, and it can occasionally burst. Can burst. Now this is going to look a little different for a plant cell. Let's take a look at what a hypotonic solution would look like in a plant cell. And here I got to go green, don't I? All right, now remember, plant cells have that rigid cell wall, and inside of it is the flexible cell membrane. Now let's say we have a plant that, I don't know, maybe it's just one particular plant type, some species of plant that has a relatively high sodium concentration inside. And let's say you put it in an environment where there's a lower concentration of sodium outside the cell. So low outside, high inside. Water's not going to exit the cell. Water's going to enter the cell. H2O will enter the cell. And the plant cell will look pretty puffy, but it's probably not going to burst because you've got this rigid cell wall here that holds the contents in place. So this actually is harder to see underneath the microscope. It ends up looking kind of like this. All right, we're back. Sorry about that. One of these days I'll get this all down and remember to put it on airplane mode and turn things to landscape and make sure that I have the microphone on. Ooh, I did that once before. We had a snow day, this was last semester, and so I recorded, because I had to make it up, so I recorded a lecture just like this, and I spent, 70 minutes talking and talking and talking and recording all this stuff, and I realized I didn't turn the microphone on, so I had to do it all again. Oh well, I'd rather forget to put on airplane mode than to not turn on my microphone. Anywho, enough water is going to enter the cell that this cell membrane is going to fill up with a lot of water. So it's just going to look puffy, really. A plant cell will just kind of look puffy in a hypotonic solution. But what's interesting about that is that still, remember, plant cells have that big central vacuole. Central vacuole. I'll just put central vac in there. And that basically allows a lot of the water that enters the cell from outside, the water can go up the central vacuole and actually travel up a plant structure. So a plant cell is probably not going to burst because the central vacuole will allow the water to be driven up through the plant structure. So plant cells have a different way of handling a hypotonic solution. All right, so we did hypertonic. Hypertonic back here, where the sodium concentration outside the cell is greater than the sodium concentration inside the cell. Water will leave the cell will dehydrate. We did hypotonic, where the sodium concentration outside the cell is less than the sodium concentration inside the cell. Water enters the cell could swell, it could bloat. There is a third option. Let's see, what if we have a cell? Let's go back to animal cells. And let's say that the sodium concentration inside the cell is the same as the sodium concentration outside the cell. Right, that is called an isotonic solution. And it's where the sodium concentration outside the cell is equal to the sodium concentration inside. There we go. And what happens here is, well, there's water inside the cell in the cytoplasm, and there's water out here in the cellular environment. And the thing is, water will basically exit the cell at the same rate as water enters the cell. So water enters and exits cell at the same rate. So we'd say that the cell is at equilibrium. Water enters the cell, it exits the cell, same rate, no big whoop, cell's at equilibrium, so nothing really interesting is happening there. But I wanted to let you know that equilibrium is a really nice thing for the cell. That's what it's always trying to achieve anyway. So there's no concentration gradient, so you're not going to have an overall movement of water in any particular direction. So that's isotonic. So for the next exam, which I need to take a look at the calendar again, because we're a week off now too. So we'll have to reevaluate when that next exam will be. But you're going to need to answer questions and know what happens in animal cells and in plant cells if you put them into a hypertonic solution, a hypotonic solution, or an isotonic solution. Please be able to know which way water moves, and please know that water moves through the process of osmosis. There, oops, I misspelled osmosis. Know the definition of osmosis. It's a little different from diffusion. It's the diffusion. It's a type of diffusion, isn't it? It's diffusion of water across a semi-permeable membrane in order to equalize a concentration gradient caused by a solute. In this case, it is sodium. All right. And I want to point out, too, that osmosis is still passive movement across the membrane. In other words, it takes no energy for water to do that. No energy is required for diffusion or osmosis. If we look way back here in our notes, way back here, when we first started talking about diffusion, passive transport versus active transport. We talked about how passive transport is passive because it doesn't require energy, because it's moving a molecule down a gradient. It's equalizing a concentration gradient. So it's like moving down a hill. Like this hill here, we have high elevation at the top, low elevation down here, and you all know it's really easy to go downhill. It doesn't take any energy to go downhill. So it doesn't take any energy to equalize this concentration gradient caused by these dye molecules here. And it doesn't take any energy to equalize this gradient caused by sodium. Water just moves automatically, dilutes out that sodium, and it doesn't cost the cell any energy. No ATP needs to be used up at all. All right. And likewise, in our hypotonic situation here, where there's a higher sodium content inside the cell than outside, water can equalize that gradient and ease the situation by moving right into the cell. Let's get rid of that. By moving into the cell, and it doesn't take any energy to do that. That's because it's like going downhill. All right. So let's talk about uphill, downhill again. All right, a concentration gradient, as you recall, concentration gradient. I'm going to abbreviate the word concentration. All right, so a concentration gradient is having a difference in concentration of a solute from one area to the next. Having a difference in concentration of a solute. And our example there was sodium. Yeah, well, we've used oxygen and other things for that too. From one area to the next. Remember that? But they're all different kinds of gradients, and we talked about an elevation gradient. Elevation gradient. So here we have high elevation at the top of the hill, and there's low elevation down at the bottom. And we talked about the ball. Let's roll a ball. Here, we haven't done yellow in a while. You know that it takes 0 energy to move a ball down a hill, right? Really, really cool. No energy at all. It's actually turns out to be a pretty cool thing. But I want to explain something here within this context. Think of it this way. When there's a ball at the top of a hill, and you just barely tip it and it rolls down to the bottom, isn't it going to go a long ways afterwards? Right? And doesn't it take some extra energy to go basically on a flat surface, let's say? What I'm pointing out here is that not only did it take no energy for the ball to go down the gradient, but it actually gained some momentum. It gained some energy in doing so. This is an important point, and I'm going to pick, I'm going to go back to yellow here. And I want us to add this to our notes here, that a concentration gradient, okay, you know it's not stable, you know nature wants it to be equalized. When a concentration gradient is equalized, just like a ball going down the hill, energy is released when that concentration gradient is equalized. All right, so I'm going to put little gold stars all around this where we write it. Energy is, and I'm going to put usually, released when a gradient, not has been, I guess, when a gradient is equalized. This is a super important sentence, so in your notes, and I hope you're still taking notes and not just watching mine, you put little stars and swirlies around it because you're going to need to find that sentence later on. It's going to become very important in a later chapter that we remember that energy is released when a concentration gradient, or actually any gradient, is equalized. So not only does it take no energy for the ball to go down the hill or your car to go down the hill, but you actually gain energy at the bottom. Right? That ball is going to go quite a ways. I mean, if you started on a flat surface and only had a flat surface for a ball to go on and the ball's just sitting there, it's not going to go far, right? You'd have to really give it a good push. But if it has gone downhill, it's going to actually gain some energy. Right, so let's remember that. All right, let's see, can I erase this stuff here? Yeah, let me draw this a slightly different way too. I'm going to need more space. Let me put that ball back there. All right, let's draw this hill again, just because it is so much fun. All right, so here we have an area of high elevation, an area of low elevation. Sure, got that. Yeah, we got a ball going down. High elevation, low elevation. This is not a concentration gradient, but kind of think of it as one because it's really similar. It's an elevation gradient, sure. You know it takes no energy to go down the bottom of the hill, and you actually know that energy is released when that gradient's equalized, right? But let's say the ball stops. Stop that ball. The ball runs out of energy at some point. Well, let's say what's up next in our ball's journey here is that there is a hill where the ball has to go from the ball or your car or you or whatever has to go from an area of low elevation up to an area of high elevation. Can you walk uphill? Can your car go uphill? Is it possible for a ball to go uphill? Is it possible? Yes, of course it's possible. We've all walked upstairs. We've all driven. Many of us have probably driven or ridden in a car or vehicle or bus, whatever, or bike up a hill. And of course you know that we humans can do that, but it takes energy. It takes energy for your car to go up Mountain Road, right? So while it takes no energy to go down, no energy required to go down, it takes energy to go up. Likewise, now let's think about let's think of this in terms of concentration gradients. What if you had an area of low concentration of a substance down here and a higher concentration of it up here? Do you think that it's even possible, or can you think of any reason why we would want to make something more concentrated? Or think of it this way. I'm going to scroll back real fast, so watch your eyes if this drives you nuts. Here we go. Here we are. This is back to our glass of water. And all these dye molecules moving from an area where they're highly concentrated here in the dye blob, and I'm doing little dots here so you can kind of see this here, see what I'm pointing to. And they're moving out here to an area of low concentration, until they're all spread out. Remember we talked about that? That's diffusion. It doesn't take any energy. That's like moving it downhill. Can you imagine all of these dye molecules somehow clustering back up and forming a blob again? So kind of like this in reverse. I'm drawing the arrow in the opposite direction now. Have you ever seen that? All of these dye molecules out here suddenly clustering in the middle? Doing that? They're not going to do that spontaneously. That would take energy, and that just doesn't happen by itself, really. Or does it? Or can we help that process along? Sure, we can. I'm going to erase this so that it's not all messy. All right, let's come back down to the bottom here. Where did we leave off? Think about what happens in Massachusetts. Well, maybe not this March now, because the world is crazy right now, as you all know, but late February, early March, something very special happens in our area. Let me see if I can find a nice, I'll use this. There's this thing that's really popular out here in Massachusetts. And we do something to our trees, and we get something very, very yummy out of certain trees. If you haven't figured out what I'm talking about, what happens, it's called maple sugaring season. Maple syrup. Maple syrup is made from the sap of a maple tree. And you've probably seen this. You've seen those little silver buckets that hang from those trees there. And people collect the maple sap, which, by the way, is very delicious to just drink straight up. It's like this bit of sparkling water that has a slight maple sweetness to it. It's really yummy. But what we do is we take the maple sap, and to turn it into syrup, think about what we got to do. We got to concentrate the sugar molecules in there. And we can do that through, well, basically, you boil the maple sap. Boil the sap. Right? So that's kind of like taking our glass of water, with our dye molecules out here, and concentrating it to make all those dye molecules in a concentrated solution. So we got to boil off all that water, right, to get our syrup. That takes energy. And that is like going uphill. We're having this low sugar concentration, and we're making a high sugar concentration there. We can do it, but you gotta go uphill, meaning you gotta expend some energy. And to make maple syrup, that's going up a concentration gradient, and we can do it, but we have to put energy into it. And what we do is we put thermal energy into it. It's energy to boil water. You can also do reverse osmosis, but that takes a lot of electrical energy to run a reverse osmosis operation too. But making maple syrup, that's like going uphill. It's going up a concentration gradient. That is not passive movement. That is active transport. Active transport in the cell. It takes energy because it moves a molecule uphill. uphill, or a better way to say it is up a gradient. Up a gradient. So it's like moving a molecule uphill. And it takes energy to move something uphill. But sometimes you gotta do it. Think of it this way. Let's say you have a cell. Let's go with a different color. We've got a cell. Here's the nucleus. I'll put the nucleus over here this time. And let's say, I don't know, there is some waste product. Let's say this molecule is a waste product. Okay, right now it looks like we have a high concentration out here and low out here, but okay, let's move 1, 2, 3 of these out. So it's 3 to 2. How are you going to move this one more out? Wouldn't that make the gradient worse? You know, doesn't one of these want to flow back? So think of it that way. To get waste products out of a cell, sometimes you have to move things against a gradient to get this last one out here, like that. Move that last one out. That sort of thing. So that's active transport. Oops, I did not realize I was still, there we go. Perfect. All right, another example of active transport besides waste products. So let's see, that's right, example, getting waste products out of cell. Another example is called the sodium potassium ion pump. Now I know you know sodium can't diffuse across the membrane, but sometimes the cell needs to move sodium across the cell membrane. So let's look at it this way. I'm going to draw this a little bit bigger here. So let's say this is outside cell, and here's the inside of the cell. I don't want to draw the whole cell because we got to do a slight close-up here. And let's say there is a bunch of sodium outside the cell and some sodium inside the cell here. There is a molecule, a protein, stuck in the cell membrane. Let me draw it like this. Let me see. There's part of the protein there. Oh, it is just so tempting to spend all this lecture time drawing pretty protein pictures. There we go. I figure I might as well make these look nice. And the other half of the protein, let's make it look like this, let's say. Here we go. So this thing in green here, this is the, what we call the sodium potassium ion pump. It's a protein embedded in the cell membrane, and you know that the cell membrane is a phospholipid bilayer. Right? Let's see, what color was I using here? Sodium. Okay, let's say the sodium ions are shaped like that. They're circular. What the heck? 3 of these can fit right in here. And as they bind there, this whole protein, how can I show you that? What if I drew it more closed like this? 3 of these sodium ions can get pumped out. So these are sodium plus ones. So there we go. But that takes ATP. So let's see. ATP, which you know is energy in the form of a cell, it's energy currency of the cell. It's got those 3 little phosphate groups. And when that initial one is chopped off, bye-bye, energy is released. That energy is used to make this sodium potassium ion pump open up and spit out those 3 sodium ions out here. So ATP is used up. But here's something interesting. Okay, so 3 of these got out here. Can you see that now there's a higher concentration of sodium outside the cell than inside? So yeah, got this wonky thing going on here. Meanwhile, no, actually, oh, that messed up my sodium potassium ion pump. Let me fix that. Meanwhile, this thing, let's say it opens up to the outside, those 3 sodium ions get out. There we go. For this thing to open back up to the cell, what happens, what color shall I use? This one. There are potassium ions, and let's make them triangular shaped. 2 potassium ions hitch a ride inside the cell. That makes this sodium potassium ion pump open up to the inside of the cell, and these potassium ions come on in. So we've got a couple of potassium ions in. All right, so it's a trade. So here are the details for sodium potassium ion pump. ATP is used to move 3 sodium ions out and 2 potassium ions in. Does that seem like a fair trade to you? We just moved 3 +1 charges out and moved only 2 +1 charges in. That is not even Steven, is it? Okay, so we've got an extra 3+ out here and a 2+ in here. Can you see that that's a charge differential there? That the sodium potassium ion pump and the fact that it creates a charge potential, that it's actually less positive on the inside of the cell membrane than it is on the outside of the cell membrane, contributes to something that we call membrane potential. It's that uneven distribution of charges that forms across the cell membrane. Uneven distribution of charges across the cell membrane. I honestly don't know what to do about this little horizontal line going across here, but whatever. Helps somebody keep track of page numbers, I guess. But that's the way Notability works. All right, but this membrane potential, where you get an uneven distribution of charges across the cell membrane, this actually gives the cell a measurable voltage across the cell membrane. So you get a measurable voltage across the membrane. And that's part of what allows a nerve impulse to travel from cell to cell. Part of it. So this is part of what allows a nerve impulse to travel along a nerve network. Just thought I'd throw that extra tidbit in there for you, for anybody who noticed, like, wait a minute, this isn't even. Pumping 3 positively charged particles out and only 2 positively charged particles back in. Yep, it contributes to something called membrane potential. Very interesting stuff. Alrighty. Now so the types of transport across the cell membrane that we've looked at have been, either categorized as passive or active. And for passive, we talked about diffusion and osmosis. For active, we talked about, waste products and the sodium, potassium ion pump. There's another form of active transport that I want to talk about that is bulk transport across membrane. Bulk transport. Transport of like really big molecules or several molecules at a time. One of these you have already seen. Let's see. So way back when we were talking about parts of the cell and we talked about, God, I always put the nucleus in the same place, don't I? I really ought to change things up. But we talked about the rough endoplasmic reticulum and proteins being made there and then being sent to the Golgi. So if this is the rough ER down here and the Golgi up here, how things travel through vesicles. Let's say, not yellow, let's make it orange. Say a little protein is made here and it gets packaged up in a vesicle that gets absorbed by the Golgi, and the Golgi will form another vesicle around that protein that it modified and then sends it to the cell membrane where it fuses with the cell membrane. And then, oops, where's my, where did I use that? There. And then a little hole is pinched in the cell membrane. So now our little protein is outside the cell. That's a form of bulk transport, and it's called exocytosis. Exocytosis, exo means to go out. So exocytosis is releasing or let's say excreting large molecules from the cell. And it requires a vesicle, requires energy, and it requires a vesicle. You've seen that before. But I want to show you the opposite of that now. The opposite of exocytosis, the opposite of exo is endo. Endocytosis is bringing in large molecules into the cell. Bringing in large molecules to the cell. Endocytosis. And it can be done a couple of ways. Amoebas will form pseudopods around things. So, I'll just use this. So, I don't know if you remember your blobby looking amoebas. This is our amoeboid cell here. And here these are pseudopods. Here, that's a pseudopod there. I misspelled pseudopod up here. There we go. Let's say there is some yummy large food particle right there. A cell will use these false feet, these pseudopods, to surround this food particle with membrane. Let me move this right there. So eventually it can pinch off. It does not like that. There we go. Right here. How am I going to draw this? Until it brings in a food particle in a vesicle. So this is endocytosis. And specifically bringing in like a large solid particle when you use pseudopods, it's called phagocytosis. Another one is called pinocytosis, but you know what? Screw this. Let's just call it endocytosis. I'm not going to make you learn these two terms there. One is just for particles in solution, one is for solid particles, but let's just call it endocytosis. Endocytosis and exocytosis. Bulk transport across the membrane. Alrighty. So I think we need a new color here. We have a new section coming up. What haven't we used yet? Green, gold, I'm kind of limited here. Let's go for a bright blue instead. All right, so we're into the section that I like to call cool stuff that cells do. And they let things cross in and out of the membrane. That's kind of cool. Another cool thing that cells do is that cells make energy. So this whole next section, this whole unit that's up next is all about cells' energy and how cells make and use energy. It takes energy to do so many different things that cells do, and so there are a lot of different molecules that are required for this and a whole bunch of different concepts. And one way that I like to start is just to talk about energy in general. And there are just some observations in science about how matter behaves and how energy works. And if you remember what an observation is in science, thinking back to that very first day, this is what I'm talking about with a scientific law. Remember, a law is just an observation. And Newton made several observations, and we actually call his laws the laws of thermodynamics. And there are and in thermodynamics, this refers to energy. You could almost call them the laws of energy, really. And there are several, but there are a couple that we're going to be using in how we explore and talk about how cells make and use energy. And the first one would be the first law of thermodynamics, which basically states energy. Energy can neither be created nor destroyed. Energy can be neither created nor destroyed. Which sounds really, really fancy. So I want to give us a more workable translation or a more workable application of what the first law of thermodynamics means to the cell, or means to how cells make energy. Right? And to demonstrate that, I want to show you one thing. Let's not go green, let's go brown for a little bit. When we talk about how cells make energy, we're going to be talking about a lot of chemical reactions. So, for example, there is a chemical reaction that happens in all of your cells, which takes a substance called C6H12O6. Ooh, does anybody know what type of molecule that is? Think about it. But we take this in and combine it with the oxygen that we breathe. And okay, we breathe in oxygen, what do we spit out? Very good, we spit out CO2. And actually, water. Okay, ding, 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 extra points if you knew what this was. It is a sugar because it's got a 1. It's a carb, simple sugar. It's got a 1:2:1 ratio of carbon, hydrogen, and oxygen. Get rid of those. And this is actually glucose. Right there, yeah. But there is something that is bugging the crap out of me with this little chemical reaction here. Can you take about 3 or 4 seconds and think about what the heck might be making me twitch a little bit? Because there's something here that is really bugging me. Do 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 dum bum 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 bum. I know that was really annoying of me to do that, but I gotta amuse myself a little bit here. The social isolation is obviously getting to me. Anywho, if you said, hey, wait a minute, there are 6 carbons over here, C6, but I only see 1 over here on this side. Okay, if these are the reactants over here and these are the products on the right, how can we start with 6 carbon molecules and only end up with 1? What happened to the other 5? Did they get sucked into a black hole? Why didn't we say something about them being sucked into a black hole? And if you notice that is excellent, because this whole energy can be neither created nor destroyed. It means for us that you can't put 6 carbons into a reaction and not get 6 out. So I gotta do something here. I gotta put a 6 there. Now we're getting 6 carbons on this side, and we have 6 over here. But wait, now I'm also noticing that there are 12 hydrogens over here, but only 2 here. What if I put a 6 there? Now we got 12 over here and 12 over here. Yay, but ooh, our oxygens are off. I've got 6, 7, 8, but I have 12 plus 6 over here. I gotta do something. Don't worry about it, I'll just put that 6 right there. That fixes it right there. What the first law of thermodynamics means to us when we're looking at a cell or any sort of chemical reaction, whether it's happening in a cell or a test tube, is that what goes in to a chemical reaction's got to come back out. You can rearrange all the atoms, but whatever atoms go into it got to come out somehow in the end. You can't make something from nothing. And atoms don't get sucked into black holes. Or if they do, you got to write it down and show it somewhere in your chemical reaction. So what this means for us is that what goes in to a chemical reaction, and I'm just going to abbreviate chemical with chem and reaction with RXN. What goes into a chemical reaction must come back out, even if it's all rearranged. So that's our first law of thermodynamics, and I am not going to make you balance all these equations. You're not going to have homework like that at all, I promise. I just wanted to point that out, why we're doing that, why these 6s are here. It's because we got to show that in order for your cells to break down 1 molecule of glucose, it takes 6 oxygens to do that. And that you're going to spit out rid of some of this mess here. And that for every 1 molecule of glucose that you spit out, that your cells take in, didn't like that, you're going to spit out 6 molecules of CO2 right there. So this molecule of glucose that has 6 carbon atoms, you're going to have 6 CO2 molecules at the end. That's the first law of thermodynamics. There. Okay, the second law, sometimes called the law of entropy. Oh, basically it states something fancy. I'm not even going to write this down. But basically everything is heading towards greater entropy or disorder. What it means for us, I'll just spell it out right now, is that a concentration gradient isn't stable. Concentration gradient isn't stable. And energy is released when it has been equalized, or when a gradient has been equalized. Excuse me. And why, yes, I actually did cough into my shirt sleeve. All right. So we got the first law of thermodynamics and the second law of thermodynamics. There we go, right there. Concentration gradient isn't stable. Energy is released when the gradient has been equalized. Alrighty. Okay, so we are running out of time there. What we will be looking at next time, because let's see, this was our this was for Tuesday, first Tuesday in April. Next time we will be picking up and we'll talk about types of chemical reactions and enzymes. And I'll see you next time. Bye-bye.